This video is going to look at food tests, which form an essential part of the practical assessment of the course. And basically need to be able to know how to do them and to do them well. The first test we look at will be the Biuret test for protein, then we look at the Benedict reagent for reducing sugars, iodine test for starch, emulsion test for lipids, and then the DCPIP test for vitamin C. The Biuret test tests for the presence of protein in a particular sample. You add your sample to a test tube, and if it's not liquid, you will need to add some water to dissolve some of the proteins. You then take some sodium hydroxide, also labeled as Biuret A, add it to your test tube, and add an equal volume of copper sulfate or Biuret B. Now, if the solution turns lilac, protein is present. If the solution does not turn lilac, so if it stays kind of a light blue color, there's no protein present. The Benedict test for reducing sugar, and like the protein, you add your sample to a test tube and you add some liquid to the sample if it's a dry sample, so the sugars can dissolve. You take up an equal volume of Benedict reagent to the sample reagent and add it to your mixture. You then take your mixture and you heat it, and if it changes color, it will have a reducing sugar present. If it doesn't, it doesn't. The key thing with Benedict to remember is that it needs to be hot, so if you're using boiled water, it needs to be still above about 80-85 degrees centigrade. And the other part is that it can be used quantitatively, so a green color has less reducing sugar in it than a yellow color, which has less than an orange color, which has less than a brick red color. The iodine test tests for the presence of starch. You place your sample onto a dropper plate and you add a few drops of iodine. If your sample goes black-blue, it contains starch. If it doesn't, it doesn't. Again, you can kind of see it quantitatively depending on the how dark it goes and if all of the starch changes, if all of the iodine changes color. The emulsion test tests for fats. So you add a liquid sample to a clean test tube. If you have a solid sample, you will need to put the sample into some ethanol and then use that. You add a couple of centimeters of ethanol and you mix with your sample. You then add water and mix again. When you mix it, if you see a white emulsion forming, lipids will be present inside your sample, so fats and oils. DCPIP tests for vitamin C. So you add your sample to a test tube, and again you cover it with water if it's a solid. You get your DCPIP reagent, which is dark blue, and when you add a drop of DCPIP to the sample, if it stays blue, there's no vitamin C. If the color disappears, vitamin C is present. You can use a titration to work out the concentration of vitamin C in your original sample, because DCPIP and vitamin C react in a one-to-one fashion.